So in this screencast, we are going to look at the process of cell division in eukaryotes. Remember, prokaryotes use binary fission. Eukaryotes use this process. And the process can be divided into basically three main steps. There's interphase, which consists of G1, S, and G2 phases. Then there's mitosis, and then there's cytokinesis. So let's take a look at what all these different things are. So the first step is interphase. And what interphase means is it's the resting phase. In other words, this is what cells do in between divisions. Remember, cells are not continually dividing most of the time. Sometimes they're just chilling out. And so interphase is kind of like that chill out phase. So some things about interphase. First, DNA is stored as if you look at this picture of interphase, you can see that the DNA is kind of in this sort of loose, tangled, almost like a big pile of rope thrown into a mess. And you hopefully remember that that is chromatin. So the DNA is stored as chromatin, which means it's coiled around those histone proteins, but it's not quite as coiled up as a chromosome yet. So the DNA is stored as chromatin. And then the interphase can be subdivided into several stages. So G0 or G0, this is the non-dividing phase. So this is where cells simply do their thing. So if a cell is not actively dividing, we say that cell is in G0, and it's just being a cell, living its life, not needing to divide. Some cells pretty much never divide and are always in G0. Examples of these include the neurons that are cells that make up your brain and spinal cord, and muscle cells, which kind of look like these long spaghetti noodle things. And you may have heard that if you injure your spinal cord or damage your spinal cord, you can't repair it. And that is true. And the reason why the spinal cord can't repair is because its cells don't divide. They're in G0. And so if that cell gets damaged, that's it, game over. So kind of interesting. When a cell sort of makes the commitment that it is going to, in fact, divide, and we will look at how a cell makes that decision in a later screencast, then it enters what is called G1. So G1 is what is called the gap phase. And in the gap phase, the cell actually grows in size. Even though G1 stands for gap, I think of it as growth because the cell is actually growing larger. Phospholipids are adding to the membrane, new organelles are produced, and then remember the mitochondria and chloroplast divide by binary fission. In other words, if we're going to divide the cell, we have to like double in size first so that when they divide, each new cell is the same size as that original. Then we begin the S phase. So S stands for synthesis. It is the DNA synthesis phase. And what happens is that the DNA as chromatin is copied. So at this point, I want to have you draw these circles, and then I'm going to turn it over to our correspondent on the ground, Mr. Sanger, who has a brand new technology tool that he would like to show off.
I'm really excited to try out my brand new webcam. This cost me about $1.49 worth of soup cans, and the best thing about it is when I'm done with my webcam, I can use it to make an amazing Tuscan white bean soup, vegetarian, good for your health, good for your environment. Anyway, we're going to start with the interphase, and you'll notice that we're going to draw a cell where 2n is equal to 4. What that means is that there are 4 chromosomes in this cell, and there are 2 copies of each chromosome to make the 4. So there's 2 different types of chromosomes, 2 copies of each gives you 4. Now, in interphase, the DNA is actually stored as a form called chromatin. And so chromatin, if I just kind of drew what it would actually look like in a microscope, I'm going to use this blue marker and then this kind of red marker. This is what the DNA would look like. It's just kind of this tangled mass of DNA, and you can't really see what's going on in there. And so for our purposes, we want to be able to track these individual chromosomes, these individual strands of DNA to understand what's going on. So what I'm going to do is I'm going to use a squiggly line to represent a distinct thing that's eventually going to become a chromosome. And so the squiggle is to kind of represent, just kind of remind us that the DNA is actually stored as chromosome while still allowing us to track numbers. So if 2n is equal to 4, you're going to have 2 copies of each chromosome. So you have this blue long one, and you have a red long one. You have a blue short one, and you have a red short one. So basically there's two types of chromosomes, the long and the short, and there's two copies of each. One copy you get from your dad, one copy you get from your mom, and the total number of chromosomes is 1, 2, 3, 4. So 2n, two copies of each chromosome, equals 4, and n in this case is equal to 2 to make the math work. Okay, so in interphase, we start like this, but then during the S phase of interphase, that DNA is replicated. Now, we're going to talk in great detail in a later unit about how that DNA is replicated. For now, we're just going to be like, you know what, magic. It just kind of happens. Trust us, we'll get to the details later. But when the DNA replicates, that little line is going to become an X. Line becomes an X. Line becomes an X. Line becomes an X. Now, here's where a lot of kids get thrown off. In this cell, 2n is still equal to 4. And kids are like, wait, but there's like more DNA here than there was. Shouldn't we double something at some point? And no. And the reason why, there's a couple ways to think about it. You can think of it one way, that we haven't really created new chromosomes. We've copied existing ones, but there's not any new information here that wasn't there before. These are just replicated chromosomes. So I like to think of these as chromosomes, and I kind of say that these are replicated. Another way to think about it is that we count the centromeres, that little dot in the center that kind of holds them together. And so here you have 4 centromeres, and here you have 4 centromeres. And so 2n is equal to 4 in both cases.
Okay, so just to recap some things here, in the S phase, the DNA is replicated, as Mr. Sanger discussed. And so that, of course, gives us some new terminology. So sister chromatids are 2 identical copies of the DNA linked together on the same chromosome. Okay, so here were the original chromosomes, and what happens is when it's copied, it forms an X. Each half of that X is a sister chromatid, and that is identical information. The 2 sister chromatids are identical. The centromere is the structure that sort of holds those sister chromatids together. Another term I'll just mention here, it's not in the notes, but homologous chromosomes is the term for the 2 non-identical copies of the same chromosome. So the red and the blue that Mr. Sanger was just discussing. Okay, so after the S phase, we enter the G2 phase. G2 is where the cell prepares for division. So something called the centrosomes replicate. Well, we talked about that. The centrosomes replicate, and then we begin to divide. So at this point, interphase is over, and then the cell begins what is called mitosis. And you can see in terms of how much time a cell spends in each phase, mitosis is a relatively short amount of time compared to all these other areas. So mitosis is where the sister chromatids separate out. How does that work? Well, it begins with prophase. Think prophase, prepare. And a number of things happen. The chromosomes condense, the spindle forms, the nuclear envelope begins to break down, and then the spindle fibers bind to centromeres. I think it's helpful to do a little drawing, so let's turn it over to Mr. Sanger for that.
For prophase, a couple things happen. The first thing that happens is this chromatin, this kind of messy, tangled mesh-like stuff, it actually condenses into an official chromosome. So now instead of those kind of squiggly X's, we're going to make nice, solid X's. So the chromosomes have condensed. The chromatin is now a chromosome. Okay? The other thing that happens is something called the spindle starts to form. The spindle are these protein fibers, and at the ends of them are these little Coke can-like structures called centrioles, and then that spindle is just going to kind of be these little protein threads that radiate out from there. Okay? The other thing that starts to happen in the middle of prophase, I'm not really showing this in my diagram, but the nuclear envelope, this is all happening in the nucleus, starts to fall apart.
And we're back. So metaphase is next. Metaphase, think middle. Metaphase, middle. And here the chromosomes line up down the center of the cell. let's have Mr. Sanger draw it out for us.
Between prophase and metaphase, the spindle fibers actually attach to the centromeres of the chromosomes and starts to sort of begin this tug of war process and slowly begins to move those chromosomes. And in the process, essentially gets them to line up down the center of the cell. And so in metaphase, I'm going to draw my chromosomes lined up down the center of the cell. Now note that the order is totally random. I went blue, red, but you could have gone red, blue, blue, red, red. It doesn't matter. It's totally random. And again, the driving force behind them moving to the middle is that the spindle fibers attached to those centromeres and sort of created this tug of war until they were lined up down the center of the cell. So the way to remember this is metaphase, think middle, because they're lined up down the middle of the cell. By the way, prophase, think prepare, because it's preparing for cell division by condensing the chromosomes and building that spindle.
And so here's just another diagram that looks at how that microtubule, those microtubules that make up the spindle, actually changes in length. And what happens is there's these little monomers that make up these microtubules. And what's happening is to lengthen and shorten the spindle fibers, they're either adding on monomers or removing monomers. So we kind of are changing the length of the spindle by kind of adding these little subunits and taking away the subunits. So a little side note. Next is anaphase. Think anaphase apart. So in anaphase, the sister chromatids separate at the centromere and move to opposite ends of the cell, as you can see here. Let's turn it over to Mr. Sanger.
In anaphase, what happens is the chromosomes begin to get pulled apart. So I'll start by drawing kind of a shortened spindle fibers here. And what happens is that spindle is basically breaking down and separating the sister chromatids of each chromosome. So this X is getting pulled apart like that. And so one half of the X is getting pulled that way, another half getting pulled that way. And that's happening to all 4 chromosomes. And so for anaphase, think apart, because the chromosomes are getting pulled apart, the sister chromatids are going in opposite directions.
And we're back. Telophase means 2. And in telophase, what happens is the cell begins to split apart. And you can see this too. This is a very subtly done diagram. I actually took 2 circles, overlapped them, and put a square over the top. Very seamless to make it look like it's supposed to look.
In telophase, what's happening is the spindle has essentially managed to pull the chromosomes to opposite ends of the cell. And then the cell itself is beginning to divide. And so eventually, during this process called cytokinesis, these cells are going to pinch apart into 2 separate cells. So you can think telophase 2, because that process of splitting in 2 is beginning. And then note that by the time we're done, we have 1, 2, 3, 4 chromosomes and 2 copies of the long, 2 copies of the short. So 2n is equal to 4 once again, but over here, 2n is equal to 4. So we started, if I could adjust my webcam here, we started with 1 cell where 2n was equal to 4, and because of this doubling and splitting, we end with 2 cells of 2n is equal to 4. So we have now successfully made 2 identical copies of that original cell.
The chromosomes at this point begin to uncoil back to chromatin, the nuclear envelope reforms, and then we begin to actually split them completely and totally. And that process is called cytokinesis. Cytokinesis is the process where the cytoplasm divides. And just be aware that the process is slightly different in plants and animal cells because of the cell wall. So in animal cells, something called a cleavage furrow is formed where proteins kind of form a ring around it, you could imagine a balloon and then kind of squeezing down the middle of the balloon. There's this protein ring that kind of squeezes it and slowly splits it apart that way. Plants, because they have a cell wall, they essentially just start to build a cell wall down the middle of the two cells. That cell wall is called a cell plate as it's being built. So basically, in either case, the cytoplasm is now dividing. So here's another little diagram through the evolution of iPhones that kind of shows that cell division business. And then note that there's a number of great video resources that illustrate this. Here's a video of actual cells dividing, which is really cool. And then here's just a number of recap videos that I think are worthwhile to click on for additional help. But the gist is, prophase, we prepare by condensing the chromosomes. They line up down the center in metaphase. Anaphase, the sister chromatids get pulled apart. Telophase, it starts to pinch apart. Here's a couple more resources which are also very interesting. And the last thing I'll say about this is keep in mind that this is a long, continuous process. So don't, try and think of it as a flow as opposed to it's like, and then stage. It's like a cartoon, not a comic strip, if that makes sense. Okay, so that's it for cell division.